Okay, thank you for coming or connecting to my talk. I'm going to be talking about the factors that regulate or we think might regulate nitrogen in tropical streams. So one of the patterns that has baffled biogeochemists for a while is the fact that tropical forests seem to have high nitrogen availability. They have high nitrogen in the soils. They recycle large quantities of nitrogen. They have high abundance of nitrogen fixing plants, nitrogen fixing trees, and there's sustained export of bioavailable nitrogen leaving these sites. So the nitrate particularly, but also the ammonium of streams draining these tropical forests are similar to nitrogen polluted watersheds in temperate areas. One hypothesis that has been proposed to explain this is the idea of a leaky nitrostat model, in that there are spatially and temporally areas that are nitrogen poor and nitrogen rich, and that leads to fact that overall you have high nitrogen availability. So there might be layers of nitrogen poor in the litter, and this leads to active nitrogen fixation. There might be trees themselves which are nitrogen poor, and so there might be active N fixation in the trunks of trees. There's also atmospheric deposition, and you combine all of these, and these sustain high nitrogen losses from these areas. But I was really interested in trying to understand if we can see a signature of the nitrogen fixing trees in the nitrogen losses, the nitrogen in the stream in a tropical site. So the questions that I'm going to address in this talk is: this higher abundance of nitrogen fixing trees lead to higher nitrogen concentrations in the streams? You would expect if there's more nitrogen fixing trees, if they're all fixing the same amount of nitrogen, all things being equal, you would expect higher nitrate in those streams draining those areas. This is more of a spatial pattern. How about a temporal pattern? Do years that the years with higher growth of N fixing trees lead to higher nitrogen concentrations in the streams? So this is more of a temporal pattern. And then how important could this symbiotic N fixation that's happening in these trees be as a nitrogen source for these tropical waters? So this research occurred in La Selva Biological Station, which is run by the Organization for Tropical Studies. There's a lot of long-term research going on in this site. I'm going to use data from two long-term projects: the Streams project, started by Kathy Pringle, which is the project I did my PhD under, and the Carbono project, which was started by David and Deborah Clark. And so the Streams project have studied the variability of these streams across La Selva. Some of these streams receive interbasin geothermally modified groundwater inputs. These lead to solute-rich conditions where you have high sodium, magnesium. They have higher phosphorus, which we spent a lot of time studying and understanding. They also have higher alkalinity, which we've also spent a lot of time looking at. So there's this natural gradient in phosphorus. But it's interesting, there doesn't seem to be any temperature effect. So Minor Hidalgo, the photographed here, has been collecting water samples. There's 16 sites. I'm going to use a subset of those sites. He collects the water samples, he filters them in the field, we freeze them, send them back to either UGA or NC State, and analyze them for nitrate, ammonium, and phosphate. I'm going to use data from 97 to 2017, but the project is still ongoing. And then the Carbono is interested in forest dynamics and the amount of carbon in these tropical forests. So they set up 16.5 hectare plots across the La Selva landscape, 6 in each of these 3 soil types: flat alluvial soils, flat residual soils, and sloping residual soils. All stems, they measure all stems more than 10 centimeters. They've been doing the annual tree census, and they do a lot of QA, QC, and have measured to 2 millimeter resolution of this DBH, the diameter of breast height. Now keep in mind that measuring the diameter in these areas is not easy. These trees have buttresses, so they have to use ladders, and there's lots of stuff growing on them, so they have to be really careful. And again, I'm going to use their data from 97 to 2017, their annual growth. And so here are the stream sites and the plots that I used. So I'm going to use the names for the streams because these are the ones, this is Society of Freshwater Science. These are the ones that we care more about. The paired plot, the distance between those, you can see that the closest one is 138 meter. The farthest one is about 486 meters, so a little less than a kilometer away. So the sites are relatively close by. I also included the discharge. You can see that we have a couple of small streams and some medium-sized one and a couple of big ones. For now, I'm combining all of them together just to see, is there a signature from the trees that we can see in the nitrate in the stream? So we look at the basal area of these different sites. Again, I'm using the stream site name, but they come from those plots. So here's the basal area in black is for all the trees. And then in gray is just the nitrogen fixing trees. So this is the total basal area. If you look at the percentage of the basal area made up of nitrogen fixers, is somewhere between 20% and 57% of the basal area is made up by nitrogen fixing trees. I should mention the majority of this comes from one species, Pentaclethra macroloba, which makes up about 51% of the basal area across all of these sites. And then there's a couple of different species from the genus Inga that are also nitrogen fixers that are common, but these all will take up 1 or 2%. So by far the most common one is this Pentaclethra macroloba, which is a nitrogen fixer and which we know is a facultative end fixer related to the amount of light that is available. So we look at the nitrogen in the streams. The black bars here is the ammonium. Again, all the stream sites. Ammonium tends to be relatively low across the sites, around 50 micrograms per liter. Nitrate is somewhere between 150 and 250 across all these different sites. And DIN, again, somewhere between 200 and 300 DIN across all of these sites. So you can see that nitrogen seems to be higher across all of the sites, and nitrate is the dominant form in these systems. And it's interesting that the interbasin groundwater transfer affects the phosphorus as shown here. You can see that some can have 250 micrograms per liter or about 100 micrograms per liter, while the streams that don't receive those interbasin groundwater transfers have less than 10. So transfers affect phosphorus a lot, but they do not seem to affect the amount of nitrogen in the stream. So if we look spatially at the percentage of nitrogen fixers basal area versus the nitrate concentration or the ammonium concentration, you can see that there's no clear relationship. So nitrate concentrations really doesn't change that much when you go from 20% to almost 60% of the basal area. And same with ammonium. It doesn't change that much. There's one that has a higher ammonium concentration. This tends to be a more swampy area. But overall, there does not seem to be a spatial pattern. We look at the growth of these, the nitrogen fixing trees across these different sites and across these years. Again, this is 20 years of growth. And you know, I'm showing you this data quickly, but it's amazing that we have this kind of data for these long and such high quality data. You can see the growth goes from about 3 millimeters to 10 millimeters. Some of the sites seem to be fairly high, like the Saltito 100. Some seem to be consistently low, like the Arboleda seep. But notice that there's no big up or down. There's no really bad year or no really good year overall. We look at the nitrate and ammonium concentrations in the streams across the sites. There appears to be a peak of nitrate around 2009, 2010, and it's fairly consistent across all the sites. There doesn't seem to be that kind of a peak in ammonium. Maybe there's an increase in the last couple of years. Again, ammonium tends to be much lower than nitrate. So if we combine those two different data sets, our nitrate concentrations with the carbonal growth rates, no relationship. So we look at the nitrate concentration here versus the annual growth. You can see, again, I could put a line through there, but it would be pretty much flat. There's no real change. If we look at the amount of ammonium, there's a slight increase in ammonium in years that the Pentaclethra or that the end fixers seem to grow more. Now, you know, There's a lot of noise. It doesn't explain a lot of the variability, and it has a p-value of 0.8. I'll let you decide whether or not that's significant. So there seems to be a little bit of a signal there, but it's not very strong. If we look at the percentage of DIN made up of ammonium, since the nitrate pretty much stayed constant and the ammonium went up a little bit, so then this proportion would increase a little bit as well. So if we put numbers together to try to understand how important could this symbiotic N fixation, what's happening with the trees, be to the overall nitrogen budgets of these watersheds. So here, all these numbers are in kilograms per hectare per year. Here's atmospheric deposition, about 4 to 8 kilograms per hectare per year. Leaf litter is about 130 kilograms per hectare per year. I won't count that as an input. That could be just internal recycling. If we look at symbiotic N fixation, so this is what the trees would be doing, somewhere between 0.5 and 2.3. And then asymbiotic nitrogen fixation, so this is what's happening in the soils away from the trees, is about 2.8. So you can notice that what's being exported from the stream is very similar to what is coming in from the atmospheric deposition. So this would suggest that maybe this internal N fixation might not be that important, and this watershed seems to be close to equilibrium. Now, this is a watershed that doesn't receive the inner basin groundwater transfer. If we look at one that does, we have about 28 to 30 kilograms per hectare per year being exported. So this is a lot of nitrogen that's leaving these watersheds. In some ways, it makes sense because there's a lot more water coming in that inner basin groundwater transfer. But when we look at the DIN concentrations in the streams, they seem to be fairly similar. So there's something that seems to be maintaining the N concentrations in the stream relatively similar across all of these streams, even though you have very different amounts of water, very different amount of phosphorus. The N fixation in these watersheds, I'm using the same numbers because this is what's available. This comes from the study from Taylor et al. They measured this in the old growth areas in La Selva, but they didn't differentiate whether or not it's either the alluvial soils or the residual soils. So I think it would be interesting to go in and measure it across these different sites to see how this might be changing and whether that could account a little bit more for that extra nitrogen that seems to be leaving these sites. And so we're going back to our questions. Is there a higher abundance of nitrogen fixing trees lead to higher N concentrations in the streams? No. Do the years with higher growth of N fixing trees lead to higher nitrogen concentrations in the stream? I'll say no, even though there's a little bit of a signal with higher ammonium. So maybe yes, but it doesn't seem to be that strong of a signal. And so how important could this symbiotic N fixation be as a nitrogen source for these tropical watersheds? We don't know yet. We need more measurements of symbiotic and asymbiotic nitrogen fixations across all these different sites. We have some estimates of denitrification happening in the stream, but there doesn't seem to be a pattern related to phosphorus either. And so we need more of these budget numbers so that we can start closing these budgets. And with that, I'd like to acknowledge Minor Hidalgo for collecting a lot of water samples, the different technicians from the Carbono project, OTS station staff. Wagner Lopez Vargas was very helpful in making that map I showed you earlier. And with that, I'll leave my questions, contact information, and I'll stop.